Dear students, Anavarakum, Vanakam, Vandanam, Namaskaram, Dr.N.Sathyamoorthy from Trichy. Today, I like to discuss 5 more questions in biotechnology, the 4th chapter in your book. Are you ready? Biotechnology is nothing but applying technology in biology and the various branches of biotechnology, genetic engineering, plant tissue culture, and protoplasmic fusion. The first question for you in biotechnology, question number 1, mention the applications of biotechnology. Where this biotechnology is used. I'd like to give a brief idea regarding this now. Biotechnology is widely used in various fields like agriculture. We get more yield using biotechnology. Medicine, we are genetically manipulating the organism to get medicine, hormones, enzymes, vitamins like that. Environment, phytoremediation to clear the environment pollution, and commercial industries for alcohol and other chemical production. And it is used in pesticide-resistant, stress-resistant, and disease-resistant varieties in agricultural crops. That means biotechnology helps to produce genetically modified organism, pest-resistant plants, stress-resistant plant, and disease-resistant plant varieties in agriculture. It helps to increase the yield to the farmers. Then the industrial bacterium E. coli, it's an example for commensals used to produce human insulin called humulin. And this humulin used to treat type 1 diabetes or insulin-dependent diabetes mellitus. The role of insulin. The main function of insulin is to maintain the blood glucose level at 70 to 110 milligram per 100 ml. If the secretion of insulin is reduced, then it leads to hyperglycemia, increase in blood glucose level. To control or to maintain the blood glucose level, doctors are suggested as to take insulin. That insulin we are getting from the industrial bacterium E. coli. The insulin is said to be the humulin. And production of SCP, single cell protein, dried cells of microorganisms or the unicellular organisms used as a protein diet for human consumption or fodder for animal. We are calling it as SCP. With the help of photo bioreactor, we are producing large scale SAP. We are getting SAP from chlorella and yeast, the Saccharomyces cerevisiae, we say. And biochip-based biological computer is one of the success in biotechnology. Now, the scientists are fixing biochips in our body. The doctors are fixing biochips in our body. And it helps to control, regulate the various metabolic activities in our system. It helps to monitor the functioning of our body and the secretion of hormones like that. Okay. And it also used to produce secondary metabolites in large quantities and production of biofertilizers, production of biopesticides, and large-scale production of enzymes using the fermenter. Secondary metabolites are the end product of metabolism, and it's of 2 types, namely the primary and the secondary metabolites. Primary metabolites are produced in large scale, large quantities, but the secondary metabolites are produced in very less quantities, and it is very useful to humans. Okay, or flavonoids, alkaloids, which are all comes under the secondary metabolites. And fertilizers of biological origin, we are calling it as biofertilizers, the cyanobacteria and the bacteria, Nosta cannabina oscillatoria, which comes under cyanobacteria, and Acetobacter clostridium, the bacterial species we produce in large scales. And pesticides of biological origin, we are calling it as biopesticides, Bacillus thuringiensis, we can produce. And the important enzymes, so helps in the industry, we can produce large amount. And biomass energy, biofuel, bioremediation techniques are done by the environmental biotechnology. Now, a new kind of biotechnology is evolved called the environmental biotechnology. It helps to maintain a proper ecosystem in the world. And then it helps to maintain the ecological balance. So we can produce biomass energy and biofuel using algae and phytoremediation, bioremediation, clearing the environmental pollution with the help of biological option. We are calling it as bioremediation, and new science from biotechnology is evolved, that environmental biotechnology. Okay, that's all about the first question. Now we'll go to the next question, question number 2 for you. What are restriction enzyme? Mention the type with role in biotechnology. Mention that type and role in biotechnology. Restriction enzyme or restriction endonuclease is secreted by the bacterial species only. Bacteria. Bacteria, bacteria. This is bacteria. And bacteria with the genomic DNA, this is genomic DNA of bacteria. Okay. The viruses infecting the bacteria are known as the bacteriophages. So this is a phage which is infecting the bacteria, which is affecting the bacteria. I had to learn the technology. That's why it's somewhat difficult for me to draw in the computer-like structure. Okay, this is bacteriophage. Bacteriophage first attached to the body of the bacteria. We are calling it as adsorption. Then it sends its genetic material, DNA, into the bacterial cell. So entry of the viral DNA. This phage means virus. The virus is infecting the bacteria, or called as bacteriophages. Then proliferation of the bacteriophage. Finally, lysis of the bacteria leads to death of the bacterial cell. Proliferation of the virus causes death of the bacterial cell. We are calling it a lytic cycle. So bacteria want to protect itself from the viral infection or viral attack. So it secretes an enzyme called a restriction enzyme or a restriction endonucleases. The main function of restriction enzyme is to cut the invading viral DNA into pieces. Now, more than 900 restriction enzymes are isolated from the bacterial species. It helps to cut the plant, animal, and other organisms' DNA into pieces, and it is very useful in gene transfer. So restriction enzyme or restriction endonucleases extracted from the bacteria. More than 900 restriction enzymes are isolated, and it has got the ability to cut the DNA into pieces. So in biotechnology, in genetic engineering, this restriction enzyme used as a molecular scissor. Okay. E. coli, the enzyme obtained from the E. coli is said to be EcoR1, the restriction enzyme name. Like that, more than 900 restriction enzymes are isolated. Based on their mode of action, how it cut the DNA, restriction enzyme is classified into two types. One is called the exonucleases, another is called endonucleases. Exonucleases are used to cut the terminal end of the nucleotide sequence. It has got the ability to cut the terminal end of the DNA, because DNA is a polynucleotide. A nucleotide, the nanoparticle of life, the nanoparticle of DNA and RNA, are chemically composed of nitrogenous base, sugar, and the phosphate. So exonucleases, the enzymes, has got the ability to cut the terminal end of nucleotide sequence. Example for this exonuclease is Bol31 and exonuclease 3. Like that, endonucleases are also secreted by the bacterial species. Endonuclease enzyme is used to cut the DNA, in the internal phosphodiester bond of nucleotide. So if you cut in the A peripheral region, we are calling it as exonuclease, peripheral region of the nucleotide sequence or DNA or RNA, we are calling it as exonucleases. If it cut in the middle or if it creates the internal cuts, we are calling it as endonucleases. Then example for endonucleases, HIND2 and ECOR1. Okay, see here, nucleases, one is exo, another is endo. Exo has got the ability to cut the DNA or the nucleotide sequence in the peripheral region or the end. And endonucleases cut the DNA or RNA or the nucleotide sequence in the middle. This is called internal cut. This is called the external cut. So you should remember the examples for the exo and endonucleases. Then three main classes of restriction endonucleases mentioned in our book. One is restriction endonucleases is of three types. One is type 1, another is type 2. Wait for a while. Three types. One is type 1. I am not adept in technology, just learning now. Then type 2 and another is type 3. In rDNA technology, in recombinant DNA technology, generally we are using the type 2 enzymes. This is very important for our examination, annual examination, as well as for the NEET examination. Then these enzymes cut in a specific nucleotide sequence, or that is the exo and the endonucleases recognize and cut the DNA in a specific nucleotide sequence. And this enzyme consisting of 4 to 8 base pairs. Okay. Okay. Then next question for you. Is there any possibilities to transfer a suitable desirable gene to a host plant without vector? Justify your answer or explain the direct or vectorless gene transfer method. In biotechnology, in genetic engineering, we need a donor and a host. Sometimes we can transfer the particular DNA fragment into the host via vector. Here the question is there any possibility to transfer suitable desirable gene to host plant without vector? Yes, possibilities are there. We can transfer the gene from the donor to the host without the help of the vector. And this is called a vectorless gene transfer methodology. Bacteriophages, plasmid, and retroviruses are used as vector. Here we are going to see the vectorless transfer in plants as well as animals for the production of genetically modified organisms or transgenic plants or transgenic animals. And this vectorless transfer possible through chemicals, or using microinjection, or by electroporation, or by biolistics. And finally, liposome-mediated gene transfer. We can see one by one, okay? First is vectorless transfer. The first one is chemical-mediated gene transfer. Chemical-mediated gene transfer, 1 minute. I take the pen to annotate. So vectorless chemical mediated gene transfer is the first one. For this process, we are using two chemicals. One is polyethylene glycol and another is dextran sulfate. It induces the plant protoplast to take DNA. So technically says we are calling it as a transfection, uptake of gene or uptake of a nucleus or uptake of DNA fragment or uptake of chromosome by a cell is said to be transfection. So for this transfection, we use these chemicals. One is polyethylene glycol and dextran sulfate. These two induce the plant protoplast or the host to take DNA. Then second one is micro injection. We are using an injection. Okay, our DNA is directly injected into the nucleus where the genetic material DNA is present, using fine tipped glass needle or micropipette to transform plant cells. You take the gene of interest or the donor. in a microinjection, it is straight away injected into the nucleus controlling center of a cell where the genetic material DNA is present. This is called a direct method. Directly, you are injecting the gene of interest into a host using microinjection. This is the second one. And the third one is electroporation method of gene transfer. Electroporation means so you place the cell in a culture medium and you introduce a high voltage current. What will happen? 1 minute. So electroporation method of gene transfer. In this method, the host cell is placed in a culture medium. If you want to introduce the foreign DNA into the host cell in the culture medium, you introduce electricity or high voltage is applied to the protoplast. What will happen? It will create minute pores. So here pores are created by using electricity. We are calling it as electroporation. And through these pores, the entry of donor DNA into the host where they are fused to form a hybrid DNA or DNA, the recombinant DNA. It's another method. Then the next one is liposome-mediated method of gene transfer. Liposome-mediated method of gene transfer. What are liposomes? Liposomes are the artificial phospholipid vesicle. Helps in the transfer of gene or DNA. See here, this is liposome. It is a phospholipid vesicle. In this vesicle, you are introducing the DNA. This phospholipid vesicle carry the DNA into the host cell where it is fused with the donor DNA and we get our DNA. So we are transferring genetic material or DNA fragment or nucleotide or gene of interest from the donor to the host using an artificial phospholipid vesicle. This is said to be liposome-mediated method of gene transfer. Okay. Then finally, gene gun method or biolistics. What is the process? The foreign DNA is coated onto the surface of the minute gold or tungsten particle, bombarded onto the target tissue or cells using gene gun or micro projectile gun. So we are introducing the foreign gene into the system by using a gene gun. If the gun carries a gene and helps in transfer, we are calling it as gene gun method. Here the foreign DNA, the gene of interest, is coated with minute gold or tungsten particles. And then through this gene gun, it is introduced into the host. This is called the biolistics or gene gun method. Okay. Here, see, this is the gene gun. Through this, the foreign gene is introduced, and this is target cell. Target cells means a host cell. The donor or gene of interest introduced into the host by using a gun. Okay. Write the advantages and disadvantages of BT cotton. You know BT cotton, Bacillus thuringiensis, cry gene or BT2 gene introduced to cotton is said to be BT cotton. Donor is Bacillus thuringiensis, the biopesticide with cry gene or BT2 gene which secrete a chemical called delta endotoxin. The gene from Bacillus thuringiensis introduced into the system of cotton through Agrobacterium tumefaciens bacteria. What are the advantages and disadvantages of this BT cotton? First, it helps to increase the crop yield. So farmers' yield is very, very important. It is beneficial because it's a cash crop. It reduces insecticide use. Already the plant possesses insecticidal properties. So we no need to use the insecticides. It reduces the cultivation cost because if the usage of chemicals or insecticides is less, then it is beneficial to the farmers and the cost of cultivation is also reduced. Then, sorry, learning technology, sorry. Then what are the disadvantages of this BT cotton? Cost of BT cotton seed is high. So seed cost is very high because it's a genetically modified one. In the lab for the production of these seeds, the production cost is more. That's why sales cost is also more. Then effectiveness is only 120 days, the insecticidal property. After the time, the efficiency is reduced. Then ineffective against sucking pest, not for all, but it is not effective against sucking pest. Affects a pollinating insect. So because pollination is a very important phenomena for seed formation. The genetically modified crop may affect the pollinating insects because the chemicals are also present in the flower. So these are all of the disadvantages of BT cotton. Then next question for you. What is bioremediation? And give some examples of bioremediation. In Tamil, we are calling it as Uiri Vadi Tiruttam. Clearing the environmental pollution with the help of biological options, we are calling it as bioremediation. Use of microorganisms or plants to clean up our environmental pollution, or clearing environmental pollution with the help of biological options, either using microorganisms or the plants. Example for bioremediation. So number one, phytoremediation. Use the plants to clean up environmental pollutant. So the pollution causing agents are called as the pollutants. So using plants, we can clear the environment pollution. That kind of remediation is said to be phytoremediation, phytoplant remediation. A second is mycoremediation. Mycology means study of fungi. So clearing environmental pollution with the help of fungus or fungi, we are calling it as mycoremediation. And third is bioventing. Increase the oxygen or airflow to accelerate the degradation of environmental pollution. So using organism, we supply more oxygen to the polluted environment. It helps to decompose the waste substances present in the polluted environment by aerobic bacteria, because oxygen is needed for aerobic bacteria. This kind of environmental pollution clearing is said to be bioventing. And then next one is bioleaching. உயிரி வழி கரைத்து பிரித்தல் அப்படின்னு தமிழ்ல சொல்லியிருக்கா. Use of microorganism to remove metal pollutant from contaminated sites. So from the contaminated water, we remove metal pollutants using microbe. This is said to be bioleaching. Then other three types are also mentioned in your book. Composting. Composting, decomposes. We will get fertilizers. Decomposes fungi and bacteria are very good examples for decomposes. The decomposes the saprophytic organism, which has got the ability to convert a dead organic matter into inorganic substances. So solid waste is composted by the microorganism into manure. This is said to be composting. The compost helps in the growth of plants because it is rich in nutrients. And rhizofiltration, using the rhizosphere of the plant. And rhizostimulation, we stimulate the rhizosphere using microorganisms. We are calling it as rhizostimulation. Rhizosphere, the root zone, Then next question for you. Write the benefits and risk of genetically modified food. Foreign gene introduced organisms are called as genetically modified organisms. And if we get food from the genetically modified organism, we are calling it as genetically modified food, or the food is obtained from transgenic plants. What are the benefits and risk? There's the advantages as well as disadvantages of GMOs. High yield without pest. So you will get more yield and it is not infected by the pest. Then 70% of reduction of pesticide usage because they show resistance against pest. So that pesticide usage is very much reduced, and it reduces soil pollution problem. If we use fertilizers and pesticides in the field, naturally the soil get polluted. So it helps to prevent the soil pollution and helps to conserve microbial population in soil. If the soil is undisturbed, then the growth of natural microbes is not affected. So rapid proliferation of microbes in the soil and decomposing ability of the soil is also increased. Okay. What are the risks? Biological magnification, entry of chemicals from the environment to human system or plant system or animal system. We are calling it as biological magnification. So, if the environment pollutant enter a human system through food, it is also said to be the biological magnification. And the biological magnification affect the chemicals present in the food, affect human liver, kidney function, and also causes cancer because the chemicals are mostly carcinogenic in nature. They stimulate the cells for mutation dangerous, and rapid proliferation of cells causes cancer in our system, and hormonal imbalance and physical disorders. So chemical stimulation leads to hormonal imbalance, either hyper or hyposecretion, and it causes physical disorders in human system. And anaphylactic shock, sudden hypersensitive reaction, we are calling it as allergy. Sudden inappropriate excessive response to the stimuli is said to be allergy. It may causes allergy also. Okay, these are all the risk of the GMOs, genetically modified organisms. And adverse factors, it affect our immune system because of bacterial protein. Immune system immunity is nothing but the disease resistance exhibited by the host towards microorganisms or their product. So it may affect our immune system. The primary, secondary, and tertiary lymphoidal organs may be affected. And it may reduce seed viability. Viability means the germinating power of the seed. The germinating power of the seed may be affected. Okay. Thank you, students. Next class, we will see the next chapter, 1 mark, 2 mark, 3 and 5 mark questions. Thank you very much. Tata, bye bye. See you. Have a great day. Thank you.